Welcome to the introduction video to the Axiom Design Center. This is part one of a two-part series reviewing the Axiom Design Center. In this video will be an introduction to the workflow and the overall terminology associated with the Design Center. The second video will be a short demonstration on how to create a myDesign array using the Design Center. In this example, it will be to designing an array for use in GWAS studies studying cancer. As a customer, you have ideas for creating a genotyping array, whether this is for a GWAS study, whether it's for targeted genotyping, or follow-on validation studies. The Axiom Design Center can help you create the myDesign array, creating the markers of interest for your study. In general, the Design Center has several ways that you can identify these markers of interest, ranging from submitting gene lists or regions to the Design Center. It also allows you to filter these marker lists based on characteristics of the markers like minor low frequency in various populations. At the heart of all this is the Axiom Genomic database. It actually contains two different flavors of markers. The first set of markers are what we refer to as Axiom validated markers. What this means is that we've been able to accurately genotype these markers using the Axiom 2.0 assay. Thus, it means that we have seen good cluster resolution or good cluster qualities, and we've also seen at least two instances of the minor allele. A second set of markers that are included in the Axiom database are what we refer to as taggable markers. These markers have not been shown to work with the Axiom assay. There are a couple of reasons. The first is we haven't assayed the marker. A second option, or second possibility, is that we haven't seen greater than two instances of the minor allele. And the third option is that for whatever reason we haven't seen good cluster resolution at this point with that marker. But these markers do have good genotype information available from other sources in the public domain, and we can use that information to determine the R-squared value associated with Axiom validated markers, enabling one to tag those markers and use the Axiom validated marker as a proxy for one of these taggable markers. One thing to mention at this point also is that within the Axiom database, it includes both SNPs and insertion and deletions. And so in general, we use the term marker to refer to the collection of those. So the design process for the Axiom Design Center is fairly straightforward. It consists of identifying a set of target markers, or markers of interest, which we refer to as the target set, deciding on the tagging strategy for that target set. You can identify more than one set of markers that you're interested in, and you can use different tagging strategies for each of those. After you've defined your sort of sets of target markers that you're interested in, you submit the design attempt to Affymetrix. There's some processing that happens, and it returns selected markers. You evaluate the set of selected markers. You can refine the criteria if you like, or if not, you can place the order and create the myDesign array. When designing a myDesign array, there's actually two different strategies that one can take from the beginning. The first is you can choose to supplement an existing Axiom array plate. In this example, you can see that there's the Axiom CEU array, so you would already start with all the SNPs that are on that array, and then you can just choose to add your custom content on that. Depending on the array that you're starting with, it will determine how much space is available for your custom content. The second approach is to create an entirely new array plate from scratch. All the SNPs you want, you pick from the very beginning, and you define your own choices. So, within the Design Center, there are multiple ways that you can choose to specify your markers of interest, or the target set. The first way is you can actually specify the markers themselves using either an RSID or an Affymetrix SNP ID. Second, you can choose to specify a gene or list of genes by either using gene symbols or even OMIM IDs or MorbidMap IDs, and then all the SNPs that are contained within the bounds of those genes or markers would be included. The third is you can actually specify specific regions on the genome to be covered simply by submitting chromosome number, start, and stop. The point that I want to make here is that while you can submit markers based on RSID, it's probably advantageous to either submit them also as a chromosome position or to submit them only as a chromosome and position. This is because dbSNP, as you know, revs its content pretty regularly, and it's possible you'll have an RSID that's not included as part of the database even though that SNP is in the database, simply because you're on a more recent version of dbSNP or an older version than we are. So just as a safety precaution, I recommend submitting the SNPs from the public domain as a chromosome and position. You can choose to submit them both as an RSID and as the chromosome and position. When the lists are processed through the system, the redundant SNPs are taken out, so only one copy of the marker will be placed on the array. So in order to ensure that the markers of interest from the public domain get on, I simply recommend using the chromosome and position so that you don't end up with them missing even though they are actually in the database. There are three more less popular routes to specifying markers of interest or the target set for the array. The first is you can actually use predefined lists that Affymetrix has provided. These are associated with a couple of a couple of diseases or pathways, some known regions, and then there's also lists of markers associated with arrays, either from Affymetrix, other Axiom arrays, or historic arrays, or even from some of our competitive arrays. Second mechanism is that you can actually specify specific probe sets from pre-existing either Axiom or myDesign arrays, simply by identifying the probe set ID and the design name or the array name, and then that actual probe set will get reused on your myDesign. And the third option here is also to support the inclusion of de novo sequences for the array plate. In this case, you may have a marker of interest that is not in the validated content of the design database. You've covered it with a tag, but you still want to get that marker placed on the array. You can do that by submitting the tab delimited file with chromosome position and sequence. That marker will be tiled on the array. The reason to include this in addition to the tagging is there's no guarantee obviously that the de novo sequence will work, but we do provide that option for your study. After you've provided or identified the list of target markers that you're interested in, it's possible to refine this list based on a couple of criteria. The first is the relationship of that marker to a transcript. Is it in the CDS? Is it in an exon intron? Is it a non-synonymous marker? So you can filter, you can provide a gene list and then choose to limit the number of the types of markers that go on the array based on that they have to be non-synonymous markers or any of the other options. A second thing is you can choose to filter based on the physical distance to a transcript. You could start either with a gene list or without and say, I want all the markers within 10 kb of this list of genes. And the third and final option for refining your target set of markers is that you can choose to do it based on minor low frequency for the marker observed in our screen in various populations or in the populations ASI, CU, and YRI. After you've identified the target set of markers that you're interested in, then you need to choose a tagging strategy that you want to use. The most straightforward tagging strategy is direct selection. That means that you will select an Axiom validated marker only out of your target set. So if the target set is validated, you get it. If it's not validated, that marker is not included on the array. The second option, which is the one that I recommend for the most important markers in your list, is to do the best tag strategy. In this case, it takes the list of target markers, it goes through and selects all of the Axiom validated markers out of the list and puts them onto the array. Then it goes through and for all the non-validated markers, identifies the validated marker from the database with the highest LD, assuming it is above 0.6 in R-squared, and puts that marker on the array. So it's basically a one-to-one, an effort to get a one-to-one mapping between your target set and the selected set. The third option is to do what we refer to as greedy tagging, which is an effort to select the fewest number of Axiom validated markers to tag the markers of interest. In this case, you need to select both the target population and an R-squared threshold for that tagging. And the third and final option is to do what we refer to as physical density. And that's simply to select validated markers evenly spaced across the gene or chromosomal region until the specified density is reached, or the minimum distance between the markers is reached, or there are no more markers to select. On the right side of the slide here, you can see for a single gene, HTR3A, we've got displayed on the top is all the markers, target markers in that gene. color coded by blue if they're taggable and red if they're validated. And you can see the effects of the different selection methods as you move down for selecting the markers from within those regions. Starting at the validated, where you get all the red ones, validated plus best tag, or the greedy selection, and then by the physical density at the bottom. So basically, you've covered, by learning the ways to submit the target set of interest and the selection method, sort of how to go about it. So the Axiom Design Center web interface is the tool that you will be interacting with to do these things. The top part up here is basically the business, gives you names, projects, and that sort of thing. And then down here in this region is where you actually specify the marker groups of interest. So it's sort of two phases: defining the target set, which has 2 steps, and then it's choosing the selection method. And so we can see here the marker group piece, which is what we refer to as the combination of the target set and a selection method. You can see step 1 is where you pick any files of interest. Step 2 allows you to filter, as I said, to refine your target set of interest based on the transcripts and the markers related to them, or other features of the markers themselves. Step 3 is to actually where you selection method by clicking on the change button, and you can see there's a greedy tag, best tag, physical density, and the direct selection. Direct selection is indicated by default. So this is how you set up one marker group. But we recognize, as part of a design attempt, that you're likely to have either more than one target set of SNPs that you're interested in. This collection of genes, and then that a second set of genes. And you may not want to use the same selection method, or you may not want to use the same filters associated with them. So what we've done is we've enabled you to select define multiple marker groups as part of a single design attempt. You can do that either by clicking the plus marker group or by copying the existing marker group and then simply editing it. More details of this and an actual sort of use of this will be shown in part 2 of the video, where you can actually see me go through a hypothetical design for a cancer GWAS and use all these buttons. So after you hit the submit button for a design attempt, it goes off, Affymetrix does some processing, and then you'll get an email response that says your project has been updated and your data is available for you to look at. And so there are a couple of things that come back as part of the process after it's been processed. There's a PDF report, there's a collection of files that are available, and I'll go through those at a high level right now. So the first thing that comes back is a design summary, or the PDF report. It identifies the number of markers of interest that you specified the target set, the number of markers that were actually selected to go on the array, which we call the selected set, the genetic coverage of the target markers, how many of them were covered, and then we characterize the selected markers by minor allele frequency breakdown and population, and also by their relationship to transcripts. And these are presented as a PDF report. This PDF report is available at 2 levels. The first level is for the design attempt, which is all the marker groups together. And then a second level is more broken down for each individual marker group, so you get the details for each marker group by itself. Besides the PDF report, we provide a set of bed files to enable genomic visualization of the results, because we recognize that in some cases you want to see sort of how well your gene is covered, or if there are any gaps in it. And so we provide bed files, which there's 2 different ones. The bed files are all broken down by chromosome, because otherwise they get too large. So we provide the target markers, or those are the markers of interest, by chromosome. A second file is the selected markers by chromosome. And then they are available at both the design attempt level and for each marker group. And bed files, as you're probably aware, are compatible with the genome browsers. Shown here is loading the bed file into either the UCSC genome browser, where you can see the target markers and the selected markers, or loading the data into Chaz. And again, the color coding that I showed previously is true for the target markers, so that they will be indicated by blue for the taggable markers, and the red indicates the validated markers. Obviously, selected markers will always be red because they are validated. In addition to these 2 files, the bed files, we also provide two additional types of files. One we're referring to as the coverage files, and the second is the annotation files. These are special files that are available upon request, aren't included as part of the initial reports provided to you, but simply request them from bioinformatics services and they can be produced. The first is what we call the coverage files, and that's basically a list of all the SNPs of interest or markers of interest that were not selected or tagged. So there's one file that just simply has, here are all the target markers that you submitted that are not tagged on the array. Either they themselves didn't get on the array or they're not tagged. The second file contains a list of all the target markers, the selected markers that tag them, and the corresponding R-squared value in between them. So you can see here that there's the target SNP ID, the target RSID, the Affy SNP ID that tags it, the corresponding RSID, and then the R-squared value. It's important to note that neither the target SNP ID nor the tag SNP ID column will be unique. Basically, a target SNP can be tagged by more than one SNP, and a SNP can tag more than one target SNP. So it's not really, it can be a quite large file for many designs. And then the third file or additional file that is available are the annotation files, which is a sort of a small version of the standard annotation provided with Affymetrix arrays. It consists of the probe set ID, Affymetrix SNP ID, DB SNP ID where available, chromosome position, strand, the alleles, and then minor allele frequencies in the ASI, CU, and YRI populations. So overall, the Design Center provides an easy mechanism to select the SNPs or markers that you are interested in to put on your array. It is supported through Affymetrix bioinformatics services who are available to consult during the design process, help answer any design questions, and just generally be available for helping to support you in your process. There are flexible methods to design and submit the content. We support gene symbols, chromosome regions, disease and biological categories, SNP types, and including other commercial arrays. Customer's unique sequence, you can submit your own de novo sequence. The tool is easy to access. It's available online 24 hours a day, and it is freely available to customers through the NetFX and Affymetrix.com websites. Additional resources to support the myDesign are available on Affymetrix.com. Their Design Center itself, the link is listed here. There are other tutorials and manuals available. These include the Design Center Quick Start Guide, the Array Design Guide, and other tutorials. And if you have any more information that is needed that's not listed here, feel free to contact technical support either by phone or via the email address that's given there. I'd like to thank you for your attention, and I hope that you will join me for the hypothetical cancer design in another tutorial.